it till when? When's your test?
Tuesday.
Tuesday. However, that has to have some logistical consequences for me that I have to cover some of the other material, if at all possible. Okay. So, settle in. I'm going to finish up Chapter 8. The reason there are no review sheets for Chapter 8 is because it's tiny, very short. Focus notes will pretty much cover it. In fact, I'm going to shorten down some of it right now. All we're doing in Chapter 8 is we're applying what we learned in Chapter 7. We're applying what we learned in Chapter 7. We want to cut apart and pick a gene out that we want to duplicate or use. We would use DNA. We would use the restriction. What? Enzymes. First cut. If you want to copy DNA, you use DNA polymerase what? 3. You want to use DNA polymerase 3. If you wanted to get rid of the RNA primer and remember do some proofreading, you'll probably do DNA polymerase 1. If you have all those pieces that you picked out that you want to now make this gene, say maybe for interferon or insulin, we would use DNA that seals them together on a lagging strand. Ligase. DNA ligase. So really, you're pulling the tools out like duct tape and scissors out of what we just learned to do recombinant genetics. What they're doing with recombinant genetics is they pull out a gene that they want to mass produce a product from, and they generally, in other cases, put it into a little E. coli. Let's think about this.
I just wondered how they do that.
How do you?
What instrument do they use?
The restriction enzymes. Okay, first, that gets really involved. If you actually want to know which part of the gene you want to do, you sometimes will hunt for the product, the messenger RNA, that makes, say, the enzyme that breaks down lactose. So then they go, which is actually what we're teaching today, take the messenger RNA that makes lactose and do the reverse transcriptase. What does that do? Goes from RNA to DNA. So as soon as they have that little itty bitty bit of gene, the RNA is already selected it out for them. That's one way to find the piece they want. Then when they want that piece, they study. There are hundreds of different types of restriction enzymes. Some cut at these nucleotides, some cut at those. They look at the ends of what they want, and they find the restriction enzymes that will go in and cut. Then they pull it out and transfer it over, and they will try to fuse it into another DNA. Answer the question?
Like what instrument do they use? Like actual physical?
Almost. They may have instruments now, but it's not really so much instruments as different enzymes you place into it. Separate it out, you take it and put it on a gel, and you run an electrophoresis is what it is. You run a current in a buffer, and it separates everything out. Then they cut those pieces out by hand, and then they move those over. Now they do have machines that can probably do this, like DNA sequencers that can do that, but it all started by hand, using reagents with enzymes in it, reagents with polymerase in it, reagents with one machine. There's no machines though. But there's not a machine. Sometimes
with a microscope to see.
no microscope can see this. Microscope, they have to do it indirectly. It's very well. They're actually so evolved now. Yes. You buy a kit, and the kit comes in the mail, and some of the researchers don't even know how to work it. Okay? I mean, they know how to work kit. They don't know how it works. They just make them do what they want. Because other researchers, digital researchers, can invent a kit for them to order. But going back to the clone, it's hands-on. That's how they taught me. They taught me hands-on original DNA. And it's this white meat that you crack out of the shell and you put it in cold ethanol because it precipitates it out. You get this white, fluffy, sort of white tube. That's actually DNA. Clean everything else out, and then you got to start separating it out. Okay? All right. I sort of just taught what I'm going to go about. You okay? What are the goals for this? Let's go back to the beginning. That's a good point. How do you find the If you want this certain gene, how do you find that out of the entire DNA? That's one of the starting points. There are several ways to do this, but the one that I'm going to talk about, which I'm jumping ahead right from that list, is called reverse transcriptase. What is transcription? It goes
DNA to
DNA. If you reverse transcriptase,
RNA
uses
to
enzymes to go
DNA.
RNA to DNA. As a small little side note, the HIV virus is a reverse transcriptase virus. It starts off as RNA. It has the enzyme reverse transcriptase. When it interferes or goes in, it goes to DNA, and it flips back to RNA long effects. But for the process for genetic engineering, think about this. Say you want to make a Pegasus, and there's a gene for making wings, and it's not that simple, okay? It's not that simple. Well, if you want to look on the DNA would probably stretch from outside this building all the way to the other side, and the gene would be a little teeny sliver about that big. But how do you find that? It's like one needle in a haystack. So one thing you can do is reverse transcriptase. If an organism is going to express, it has to open up the DNA, read it, you make messenger RNA, from messenger RNA you make the protein. But you have hundreds, if not thousands of copies of the messenger RNA, all coming from that one teeny gene. So instead of hunting for the gene on the DNA, you do what? You hunt for the thousands of pieces of messenger RNA that were made from it. When you get that, you use the reverse transcriptase enzyme. It's going to go back and make the DNA that would have made that messenger RNA. You just got one in the gene you wanted. You also were hunting for maybe 10,000 or 100,000 needles in a haystack versus one. So it's easier to do that way. Does it work that easy? No, it's not that. That's the teaching and the theory, but nothing's that clean in the laboratory. Okay, then you take the gene, all right, which you now have, and you would maybe either use that one or maybe you want to cut it out of the original. If you want to cut the gene out of the original, you would look on each side and hunt for the sequence that matches that gene. You look for the nucleotides on the end, and they have patterns. ATP, GCC, they have patterns. Then by the pattern of what you want to cut, you pick your restriction enzymes. Some restriction enzymes, these are examples, will only cut between AA and TT. Others will only cut between GCCG, GCCG, and there's a whole library of them. You pull the one that you want, that will cut out the ends you won't what? Cut. You then have a piece of DNA. You take it over. You can either use transformation. If you make, say, your E. coli a competent cell by treating it with salts or chemicals, so it does what? Takes DNA up from the environment. Or you can use a phage as a vector, which will just automatically go in a jet. Or you can, well, conjugation's not used as much. Get the DNA over to the E. coli. And of course, if you're doing it in a test tube, you're not letting the organism do it, you'd have to have DNA what? Ligase to seal it in with the pieces. And it gets more and more and more and more involved. Now let's move to E. coli. Let's say you put the gene in to E. coli for insulin. What's insulin for? Sugar levels. The person probably has diabetes. Getting that from harvested organs is expensive, intensive, and any time, especially if it's a human source, you can have carryover of viruses or even unknown viruses. And to give you an example of that, did y'all hear about the case with organ transplants about a couple years ago? This is the example I'm giving is why you want to reproduce an E. coli and not take it from harvesting something. There was a case, it's been removed from YouTube, and you have some passion for this, and it's probably done because of the pain of the families. A person went into an emergency room and was drooling off, thrashing, crazy. They labeled it as drug overdose, right? He died. Well, he had become, or had written those forms to become an organ donor. So 3 people came and received different types of organs from him. One was a young man in high school who was dying because his kidneys were failing. They went in and they had him interviewed. You can't promise him how things were removed. And at first they were fine, and the 3 people would meet each other because they had something in common. They all had organs from this one person, and they were all going to live now. They were happy. Then the boy started having hallucinations. And he said he would come out of delirium and he said, "Mom, I don't know what's happening to me. It's like everything's going crazy." And then he'd go back into hallucinations, and he fell, 3 died, all 3 of them. It was carryover. Have an idea what it would be. The original person did not have a drug overdose. They had rabies. So when they transplanted the organs, along with it hidden inside was the rabies rhabdovirus. And that was very heartbreaking. I cannot imagine what these people felt like. But that's getting back to one of the parts of recombinant genetics, which is you pull the gene out instead of harvesting it from the islets of Langerhans or wherever you're getting it from, maybe whatever. You put that one gene into E. coli. Now that little E. coli is not going to do too much because it's just one tiny little cell. And what does he do though? He doubles his numbers every 20 minutes. In a very short time, you don't have a little bit of E. coli that can produce a little bit of insulin in this case. You have a huge amount. And the organism's doing the work for you. You're not having to purify, get tissues, get organs, whether from animals or something, and have to find anything to purify. Excuse me. One person asked me, well, you're going to hurt the person if you inject E. coli to the person. You're not injecting E. coli to anybody. It's producing a product, insulin, purified, pure human insulin out of that huge vat. And it can't have contaminating water. It's nature how it's produced. You cannot have contaminated viruses. You cannot have anything that could contaminate from a human being because it was produced in a bacteria. Interferon is another one that they produce this way, with genetic engineering, putting the gene for interferon into a bacterium to get massive numbers so they can get a product. Do you know what interferon does? Interferon can shut down the protein synthesis of cells, host cells, eukaryotic. And in doing so, it stops viruses from replicating. What do viruses have to do? Enter. That's where they take over. They cause the synthesis of viral parts. Well, if the protein synthesis system of the affected cell is turned off, the cell will not make viruses because it can't. Protein system's turned off. That's another thing, interferon. Another one is human growth hormone. Sometimes when you have young children, they check blood for the femur and they give some sort of ideas about how tall the child's going to be. They may or may not decide human growth hormone to get a certain height. That's the individual choice. That's not for me to say either way. You can also do subunit vaccines and into vaccinations. I'm probably going to touch the top of this as I start this. When you're vaccinated, there's 2 ways to do it. One, inject you with a living organism that's been weakened. That's called attenuated. And that's actually the best protection you can get. That's very, very good. It has an issue. It was the organism weakened by chemicals or treatments or passage through multiple hosts. But a certain percentage of the vaccine, the living attenuated organisms, will revert to original form. What does that mean? A certain percentage of them become pathogens. And they know the numbers. They know if they take the polio attenuated vaccine or something else, out of 100,000 people, 20 or 100 are going to get polio. And that's what happens. But it gives you the best protection. It also enables you to sometimes sort of vaccinate the people around you. If one of you had an attenuated virus, or an attenuated vaccine and you're shedding it, it can go to the people sitting around you, and you just vaccinated them without ever touching. So it can spread to a certain degree. What that can have damage, right? So they sometimes, or they often move to subunit vaccines, which means they take pieces of the pathogen, not the whole pathogen, and inject it to give an immune response, which means protection. Well, if you inject a piece of a bacterium into somebody, the bacterium had to be killed. You only have like a piece of the capsule going in, or a piece of the membrane, or a piece of the cell wall. Can that revert and start to the original pathogen? No, because there's only a piece of it. A piece cannot turn into a whole bacterium. Flip side to that one is it's less immunity. All these booster shots you have to get, so tetanus, let's say every 10 years, that's probably because you have a subunit vaccine. Well, if you want to make it, you do this. You do this. You find that piece of that pathogen that causes the immune system to protect you, to respond. Put the genes for that piece into who? Well, often E. coli, but it can be somebody else. The bacterium now what? Mass produces that piece to make that subunit vaccine. And then, when you purify it out, you have just the subunit. And when you inject that to a person, they cannot get the living pathogen because it's just a piece of it. Does that make sense? Questions on this part so far? Right? Now, just a little bit of this. Recombinant DNA engineering is the same thing as genetic engineering. I said we use the enzymes from everything. Transcription, replication. I said you can use transduction. You can use a virus to inject the DNA you want into a host. Excuse me. This is just sort of history. They did not do genetic engineering in ancient Rome. I can promise you that. But what they did is they picked up on certain products being produced. So they did have beer and wine, and they had all that. You know that is really from the metabolism called fermentation. They didn't know that. There are a lot of different goals. You can use this if you want to talk about just not trying to get a factory from a bacterium. You can try to get rid of unwanted phenotypic traits. Hasn't worked yet. Okay, not yet. Okay, you can get vaccines. That's your subunit vaccine. Get antibodies. Sometimes you want to inject a person with antibodies, but the antibodies will only protect them as long as the antibodies are flowing in their bloodstream. When the antibodies disintegrate, what happens to their protection? Oh, because their body's not protecting them. You have an exterior element protecting them. The antibody is probably made in a horse or a goat against the disease. Hormones would be that, what, insulin. You can also get some enzymes or human growth hormone. That'll be some of them. PCR went over last time. Reverse transcriptase, I just talked about. Restriction enzymes, we just discussed that. Vectors, a vector's in that carries DNA from point A to point B, I said. Transduction, you using a virus. Okay, mutagens. This is sometimes used, it was used more at the beginning. Way back at the beginning of production of penicillin. Penicillin is a mold fungus. The original strain only produced a very small amount of penicillin. Very small amount. In fact, the poor police officer they first tried to save died because they ran out of penicillin. They were bringing him back. He was getting well. Ran out of the antibiotic. He died. Okay? But they have been working on this original strain for a very long time. One of the first things they did to it was they hit it with mutagens. That's like a sledgehammer. That's not very precise. Probably 10,000 strains died, and nothing good happened. By 100,000 strains died, and nothing good happens. Mutagens cause increased rate of mutation. It's a shotgun effect. They're hoping that mutagen somewhere affects the production of penicillin. It's not precise. Well, eventually they got a few strains out of there by treating with a mutagen that actually had an increased production rate of penicillin. And then they kept those strains and they tried to hit it with mutagens again, got rid of the ones that didn't work, just killed them, and kept trying to get strains with higher and higher production. that's sort of a shotgun approach. Okay? That's one way you can get a change that you want. Limited chain reaction we talked about. Yes, you can locate. Don't worry about this handout. Concept maps, I'll just now talk to you or I throw it on the board. That's what I do nowadays. We talked about restriction enzymes. I am not making you learn the specific types of sticky and blunt ends. Because it gets very involved as to what are the patterns, which ones will cut where. You have to treat blunt ends differently than sticky ends. What I want you to know is what about restriction enzymes? They cut the DNA and can be used to get the piece that you want. And the other thing I want you to know about restriction enzymes is not genetic engineering, it's what they do in nature. They are in nature not for genetic engineering, but to do what? Cut non-self DNA up and keep you safe. Keep you safe. Pharmaceutical and other applications. Protein synthesis, well in this case, that's how we do it. Insulin, interferon. Interferon is not a beautiful answer to everything. Well, how interferon that's naturally produced by your cells does is this. The cell that is infected with the virus, excuse me, the cell that is infected with the virus sometimes gives off the protein synthesis from E. coli, gives off the interferon. The cell effect is gone. It needs to be destroyed by the immune system or it's going to be killed by the virus. One or the other is going to happen. It sends interferon out to the surrounding healthy cells to save them. The infected cells are already gone. The interferon goes and shuts the protein synthesis down in the surrounding what? Healthy cells. If they don't have protein synthesis, when the viruses bump into them, and even if they enter, the viruses can't replicate. That's how that works. Okay? And of course, as it says, you can avoid any sort of virus, whether it's hepatitis or other, that's with scale. Vaccines, that's what I discussed and told you that living, which means attenuated bacteria or pieces, can revert and give you the actual disease. But if you have a piece or a subunit of the virus, etc., that will never revert and give the person the disease. Now, that doesn't mean perfect. Some people do what? Don't respond well to vaccinations, and then they actually have an allergic response, which is a whole other different issue. Whole different issue, which we'll get into with vaccines. Genetic screening. You can find, say, if you have some of these hard-to-find viruses, or maybe a spirochete that hides in the body very well. Find what their DNA sequence is, which you know from the library. Go ahead, take a sample from the patient, look in the lab, and if the two fit together, since this one was bought in the store, and it means maybe HIV is there, this one from your patient matches it, it means the patient has to have HIV. It's only complementary strands bind. Right? Now, fingerprinting simply means PCR, perhaps to identify a person at a crime scene. Not always bad. You can also say that person could not have been at that crime scene. You say they were, that's not their blood, so they have to be innocent. They have to be innocent. I always think this. Gene therapy is not working. A while back, they had a person at a major university. Okay, I'm sure they're advancing beyond this, but this was a big case several years back. They decided to go in and treat him for a genetic disorder. They sent the gene in, they fused it into his chromosome, and his body decided that it was non-self. He had a massive, what? Autoimmune response, and his body attacked himself and he died. He died. There was a lot of argument and fuss over that perhaps researchers moved too fast, because what works in a test tube, and sometimes it even doesn't do that, you can't jump to a person until you're very careful. Very careful. A medical diagnosis is simply that's similar to pretty much back up here, genetic screening, to see if the content is in there. I want to go back to genetic screening for a minute. If a lady is prone to breast cancer, some people prone to breast cancer have a gene called the BRCA gene. The BRCA gene, they will take a piece of some of your blood and they'll send it off to a lab, and they'll have, of course, a complementary strand. They'll see if that gene is somewhere after they treat your DNA in there. That doesn't mean you will get breast cancer. It means with that gene present, those people tend to have a higher percentage of breast cancer occurring in their population. There used to be a flip side to this, but I think the laws have changed. They used to keep this very quiet, because as soon as the insurance companies found out you had a BRCA gene, what happened to your health coverage? Gone. Gone. So sometimes in those days, which is in the past, the doctors wouldn't even write down the results. They would just tell you. Because they're trying to protect you. They're not trying to do anything wrong, and they're not doing anything wrong. But they didn't want you to, you know how much cancer treatment costs? Astronomical. Astronomical. And that's not only the issue anymore, because all the laws have changed. Those days are long gone. Okay. This is where I'm going to go, and then I'm going to start the review. I'm not going to go into freeze resistance as much, but all that comes down to is pure water will freeze at 0 degrees Celsius. That's given. Add any solute, salt, sugar, anything, and it will not freeze at 0 degrees. So solute particles, well, think about seawater and the Antarctic. There's a lot of water down there that's way below 0 degrees Celsius, but because the salts are in there, it's still flowing as water. They actually have to get colder to freeze salt water. You can actually do something about that, but we're not going to do that. I want to talk a little bit about herbicide resistance. You ever heard of Roundup? Okay, you spray it, okay, go ahead and kill weeds. They have tried to take some of the genes and incorporate the genes into crop plants. So they can treat the entire fields, and of course with Roundup or whatever they want to be another herbicide, the crops do what? They're resistant, they live. The weeds are not resistant, they die. They're now finding the weeds around these crop fields are becoming what? Resistant to whatever herbicide they have worked with. It's always a micro. Micro is not still and static. It's always a flow, back and forth, back and forth. And sometimes you got to pick what makes the most sense at the time. Okay, salt tolerance. Let's try to give you a metaphor. Who are you tired of hearing about is a salt tolerant organism? Staph aureus. It's salt tolerant because you sweat, the water evaporates off, and you have left a layer, amongst other things, of salt. What's happening to every one of these fields out here in the irrigator? They're putting water down. The water evaporates. What is being left in those fields every time? Salt. Guess what's happening to the salt levels of irrigated farm fields? Going up, 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 up. Another cross-link of information. Nitrogen fixation, only the bacteria can do, correct? And some of them lived in association with the roots like rhizodium, some of them were free in the dirt around the roots. However it goes, only the bacteria can take nitrogen from the air and start making, well, essentially amino acids or fixed nitrogen and give it back to the plant. The plant needs that to grow. The bacteria can't take the higher salt level that does. The bacteria that do the nitrogen fixing cannot take the higher salt level. They are dying. So they are not going to supply the plants anymore with fixed nitrogen. What happens to the ability of the plant to grow? It's gone down. Well, they're trying to, notice how this is always a constant moving chess game. They're now moving out to a very salty part of our world. That's water. It's about 75% of our world. They're going to the oceans. Also organisms, they're bacteria that live in the oceans. They have to be able to take a what? Higher salt level. They're trying to take the genes that enable those organisms to survive at higher salt move it into the nitrogen-fixing bacteria around the farm plants. And therefore, at some point in time, hopefully they will keep fixing nitrogen even though the salt in the field is going up. That's not the end of it. Because the salt's going to keep going up, and eventually it's going to go beyond a level that can be dealt with. Does that make sense? It's really bad for desert lands, because how much do you have to water desert? You have to water a lot more than you would land that naturally has some sort of water on it. So eventually, salt's going to keep climbing. All right, everybody okay? Try not to drone you to death. These are actually things. Pest resistance. You ever tried to grow your own tomatoes on your own? No? Okay. If you get these little crawly things that crawl over them, and yes, I'm not knocking butterflies and moths, what happens to your vegetables very fast in the plant? They eat them. They can strip it. All right? You have nothing. Well, there is an organism called Bacillus thuringiensis. And you'll get that more in the next one. But think of it now as Bacillus T. T for the beginning of thuringiensis. It makes a toxin called, guess what? The Bt toxin made from Bacillus thuringiensis. You can buy it. You can go to a hardware store and probably buy this. It's the Bt toxin. Dust it over your plants or your flowers if they're eating that. And it kills the insects. It's a pesticide. They want to take it a step further. That's okay. You want to get it?
No, that's fine.
Okay. No, look. I make mistakes. Everybody makes mistakes. We're all here to do the best we can. Okay. I can bring it to you.
No, it's fine. I can wait.
All right. Sorry. It happens to me too. In the quietest place, I generally have something. All right. That's just Murphy's Law. We all need to find Murphy and get something like that. Okay. That's a joke. Because there was a person called Murphy, but I think he died of old age a long time ago. So, take the gene from the bacillus that makes the toxin that kills the insect. They've now inserted it into crop plants. Plants grow, they're now themselves producing the toxin. Insect comes along, eats, insect dies. Okay, well now what's showing up in some of the insects? Resistance to the Bt toxin. Flow, back and forth, back and forth, back and forth. This one, very important, nutritional value. What do most of the people in the world depend on, and they may only get one tiny little ball a day for food? Rice. Rice. What's the nutritional value of rice? Not very good. Right? So they're trying to genetically engineer rice to put in the ability to make proteins, so it gives you some amino acids. They're even trying to get it so it maybe gives you some vitamins. That way, if that's the only thing you have all day, I think it just as easily be us, you know. You at least are getting some proteins, some amino acids, and some vitamins to help you stay healthier. They have done this to a certain degree. It's not completely done. There's always more. They call that strain the golden rice. Golden rice. You know about this? Yeah.
Miss Hall's told us about it, and
There you go. You already got it. That's good. So there's someone else that's wrong with rice about to do that. All right. Does this make sense?
I eat them every day.
I like rice.
I like to know that because I eat them every day. It's not, you know,
Oh, not the rice itself. How they grow it. Okay. If when they walk in the water, you have to flood the fields part of the time. I just have to tell you this. There's a fluke worm that has a life stage that will drill straight through your leg while you're walking in the water and will infect you and give the blood flow to you. Many of the people that help raise the rice get this and it gives them anemia, so they get extremely tired. Extremely tired. I love rice. Any questions on any of this? Okay. All right, we're going to head for a review now. Is there any subject that you would like covered more than another subject? If not, I'm just going to go ahead and start. Okay, yes.
Can you go over the process?
I didn't start that, you're talking to me. Yes.
Can you go over the process of transcription and translation again?
Yes, exactly. Okay, for transcription, which means DNA to RNA, that is this handout. I'm not going really in depth into it, but if you know what I'm about to tell you, you'll be okay. Does anybody need this handout? All right. You now have 2 strands of DNA, correct? What is the enzyme we have to use? RNA polymerase. RNA polymerase links on to the double-stranded DNA. Does it copy one or both? Copies only one. It holds, the RNA polymerase just holds the other strand away. One strand is copied directly by the RNA polymerase. Now, RNA polymerase itself can open the double-stranded DNA. DNA polymerase cannot. You have to have some help there. RNA polymerase, when it starts putting in the correct nucleotides, will put thymine, instead of thymine, it puts uracil. It's not going to put in deoxyribose, it's going to put in ribose. It copies one strand, and what just puts in the nucleotides. That's pretty much what I want you to know. There is a promoter region that tells the messenger RNA to start there, but it's really about 10 nucleotides away, which is a little bit odd. But then starts reading, and you get this long strand of messenger RNA. Now, before I go any further, eukaryotic messenger RNA and prokaryotic messenger RNA. One is active from the moment it's made, transcription. The other one is not. It has to undergo RNA processing.
What is it, prokaryotic or what?
Prokaryotic versus eukaryotic messenger RNA. One of them is active the second it's made by the RNA polymerase off the DNA. The other one has to undergo RNA processing. Remember which one's which? The eukaryotic RNA process, strand has to undergo RNA process. It has 2 types of regions in it when it's made in its transcription. What are the 2 regions? Think of the gas company for expression and the intervening sequences. Exons and introns. Exons and introns. Exons and introns. Exons, E-X-O-N-S, is for expressing regions. This is eukaryotic only. Everybody okay? Eukaryotic only. Introns are intervening. Well, you're going to have to chop them up between the exons, introns, exons, introns, exons, introns. There's an enzyme that does this. What is it? There are others, but I taught you one. Splices them. Cuts them all apart. But what does to splice mean? To splice means to twist together, put together. So it puts together only one of the 2 types of units. Be sure I'm answering your question. If I'm not, you let me know. Okay? All right? Well, only one of the 2 types of units is put together. The exons or the introns? Exons, because that's the expressing regions. Now that is still involved in going DNA to RNA, so that's still somewhat involved with the transcription, though it's not the actual transcription. Transcription from DNA to RNA needs RNA polymerase, which itself links into the double strand, has to itself open up the double strand, has this promoter region where it starts making messenger RNA off of only one strand of the double-stranded DNA, rolls down the DNA. What is it doing? It's just unzipping it as it's reading it, adding all the nucleotides, comes to some sort of region that tells it to stop. Now, we still have some of this messenger RNA linked to the DNA. We got to get it off. Because like 2 sticky pieces of tape together, or Velcro, they like to stay together. What are the 2 ways I gave you for getting RNA off of the DNA template? Excuse me. 2 different ways. Okay, who knows? I'm here to help you, not to confuse you, make you feel bad, or leave you behind. One is as the RNA polymerase is reading, it can come to a region where there is a very high percentage of G to C bonding, self-termination. Exactly right. Why would a region of high percentage of G to C help the two break apart? Breaks everything off. RNA polymerase breaks off. If it breaks off,
What's
that
it
would
called?
be
Self-termination?
self-termination. But it is a place in the DNA with a high percentage of G to C base pair ratio. Why not ATT?
Double bonds.
ATT only has 2. G to C has 3. So the bonds are tighter, and it pops everything apart. The other one's like a rock. That's my metaphor. There is a physical protein that links in and pops the two apart at that point in time. What's the name of that protein? Rho. R-H-O. Rho. So one is a physical rock that pops them apart, but it's really not a rock. It is a protein called Rho. And the other is a region of a high GC, percentage of GC base pair ratio. That's the two ways to pop them off. If that makes sense. Some use one, some use another. Okay. Now, I'm going to draw the next one for it. When it comes to, now, if we're moving from the messenger RNA to the proteins, what is that called?
Translation.
Translation. Okay, if you're tired of hearing this, 3 components have to be there. Most complete, best answer, which means you have to have all 3. What are they? You have to have your template that you read, your messenger RNA. tRNA, what is tRNA? It's transfer RNA. What does tRNA do for you? It carries the proteins from the cytoplasm of the cell into the ribosome to drop them off. Well, the amino acids. Carries each amino acid one at a time from the cell and goes and delivers them into the machine called the ribosome. It's just gave the answer to it. Now, the transfer RNA from lysine will only pick up lysine. It won't pick up anything else. Because this is called an anticodon there. Let's think about translation. What is a codon? What is a codon? A sequence, a set of 3 nucleic acids. Because where are we jumping from? We're jumping from, keep this in your mind as a big picture. You're jumping from nucleic acids to amino acids. That's a huge leap. Every 3. Code for an amino acid or a signal. It can code for an amino acid or a signal. Signal might be start here, stop here. There is a thing called codon redundancy, which really means that there can be more than one codon that codes for the what? Same amino acid. That's protection against what type of mutation? Point mutation. Point mutation. If you change one of those nucleotides and it still falls into a codon that codes for, say, lysine, you still get what in the chain? Lysine. You still put in the same amino acid in the sequence, you still get the same protein. So codons. And I just told you the next one. I quit telling the answers. Because it's better if you sort of sometimes remember it. Small and large subunit protein making machine called the ribosome. So it happens, and this is longer than this, okay, the messenger RNA. You're going to have the ribosome come in and it's going to read it. Inside the ribosome, you have 3 sites, A, P, and E. What is it reading? It's reading the codons. How did I give mnemonics for A, P, and E? A is, think about the transfer RNA now. TransRNA is going to come in like a little pickup truck. Think of it like, that's wrong, it's a codon on it. It's like a piece of delivery car that only brings in one piece at a time. Okay, and each piece has a different, one might have sausage, the other one might have bell pepper. Where does the TransRNA go in first? A for accepted. The anticodon has to match whatever the codon is right there at the A site, right? Or tRNA is not going to be able to bring in this single amino acid. This whole thing's moving. Behind this, because you're making a bead of polypeptides, amino acids, there's going to be another TransRNA coming in. What's going to happen is it's going to move to the P site. P site, you have TransRNA holding on to a what? Amino acid. But now the A site's empty, what's coming in? Whatever that codon says should come in. If this is the codon for lysine, the next TransRNA that can fit in has to be lysine. Everything moves. Now you have transfer RNA, one carrying methionine, one lysine. The P site is the what site? Processing. So not the transfer RNAs, but what's going to be linked together? The amino acids. You're going to link methionine to lysine. And you're starting to build a bead from the inside out, or a bracelet of beads. By the time it gets to the exit site, yes?
On that messenger RNA, what it's picking up, you have lysine and methionine. Is it the letters that it's matching up? The
Yes.
AUG, that's what it's.
If you look inside here and say, you look inside here, there have to be codons. I'm just going to make one up. Let's say this is AUG on the messenger RNA. When you're coming in with your anticodon carrying whatever your amino acid is, this has to be complementary. So this would have to be T, what? A, C. It's the same thing. It's the same thing with DNA, same thing with RNA, complementary. A to T, G to C, except with RNA, the T is now a U. That's how they know how to link. That's how they know how to link. Okay? Now, they start linking the beads, which are the amino acids, but you can't let this thing get clogged up with a whole bunch of transfer RNAs that stay locked to it, because then you got a mess. So as it moves, transfer RNAs go to the what site? Exit site. Now, they don't get rid of the amino acid. What the amino acid's been left behind linked to the other ones. What goes floating away? The transfer RNA goes away. Then it just goes and picks up more of its relevant amino acid and comes back in and delivers it if that is the codon that shows up later. Okay? Not trying to confuse you. Does that sort of make sense?
And if it didn't match?
If it doesn't match, the transfer RNA can't link. You won't get the wrong amino acid in. You won't get the right amino acid as coded for by the messenger RNA.
You can mix RNA and DNA, like you have the T and U.
No, that would have to be you.
Okay.
Thank you. That's me. Correct. Okay. And if you do see this, bring this up to help people. If I put a strand and ask if it's DNA and RNA, can you tell me? If it has U in it, it has to be RNA. If it has T in it, it has to be DNA, and now you can mix it. Now you can. Does that make sense? Okay, that's translation. Right. I really want to get to the new material, but I don't want to just leave y'all alone. Is there any other subject that you would like to have started or go over? Everybody okay? I want to tell you or show you a couple things real quick before I do that. If you have a weekend coming, I'm not saying, don't live your life. poorly teaching something. Wow, whoops. There we go. Okay. Now that's not what I'm showing you. Okay. YouTube. And you'll have to find the correct ones because they're put microbiology. It's spelled right. Mentor, Haven, Professor Herman. Some of these videos are genetics, some are not. Now, that's one thing to improve, not only for this unit, for any unit. Go back over here, heading into PAL, and some people can download this and some people can't, but I've only known a couple that can. No, I'm not going to show you those registers. Don't tell me not looking for that. Content. Wrong links. This is a website that's still, we continue to have things added. Couple things. Up here, useful links for later. These are all YouTube videos. All you do is click on it and it'll take you to it. The lab midterm review, if you don't have to look through YouTube, is right there. That's right there. But going back up here for everything, and you're going to have a bandwidth or a broadband or a computer that can handle this. Go to microbiology class. It's free. And go. Right on. So this is available to help you. Wear earphones because it buzzes, but you can hear it with earphones a lot better.
Okay, so UV light. You have to go into the repair systems and light off the.
Okay? So I'm not abandoning you. Those are both available for what helps you. If it doesn't help you, don't worry about it. Okay? Doesn't help you, don't use it. It does help you, please use it. Okay. All right. Okay. I'm going to get a jump, not a jump, because I postponed the test. I need to start the next material. So from here on, think unit 4. Think eukaryotes and prokaryotes. I'm going to hand out a handout on some of the first organisms. I'm going to go over some of these. This is on the very beginning. Let's first talk to you on the. This is a survey. As soon as I say a survey of life, that means we're going to say, this species has this trait, this species does this. This species has this trait, this species causes this disease. Get the idea? Memorize, 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 memorize. Probably not the best way to learn, but there's no other way to do it. Like Clostridium tetani is a gram-positive rod that causes tetanus, causes a spore former, is anaerobic, that kind of thing. But before we start here, there are two major types of life. If you're talking about, I'm going to your hand up. You want to come out and check it? Yes. Come on, you sure? Okay. Prokaryotes and eukaryotes. What do eukaryotes have that prokaryotes do not? Membrane down on the wall. I'm sorry. Got it? You have to know that. Oh, it's gonna pass the ball. Oh. Okay. Let's give everybody those two. Membrane bound organelles. For eukaryotic organisms, that means mitochondria, Golgi bodies. That means endoplasmic reticulum. That means what? Nuclear membrane, a separate nucleus. They're also larger. I am going to cover those a little bit later, but I want to start with prokaryotes. If I move into the prokaryotes, much smaller, no membrane down organelles, and I'm going to start talking about, of course, the bacteria. First, we're going to talk about what some people say are the more primitive ones, the archaea. The archaea. Now, if you don't like the format of these handouts, the same information is in PAL in the context section in text form. In text form. Everything keeps getting improved every time we get some time in the summer and through the year. If you don't like this presentation or it's confusing to you, there is a text outline form in PAL of the same information. Now, archaea are not what is called true bacteria, which is what I call bacteria. Some people consider them to be more ancient, to be older. They have a different ribosomal RNA sequence as was discovered by Woese a very long time ago. Remember WOESC, dropped all life into 3 domains. They have no peptidoglycan. I say bacteria are the only forms of life that have peptidoglycan. Archaea do have cell walls, because that's what the peptidoglycan makes up the bacterial cell wall. They just have it made of a different substance, okay? Other things it says right here, they tend to be in extreme areas, high salt content halophiles. They tend to be in high acid content. Now, if something lives, this is where the common sense comes in here. If something lives in high salt or high acid, do you think it's going to affect us? Because our environment's hostile to it. So there, at this time, and you always have to say this in science, there are no known pathogens of people. Does that mean they won't find one day? They could. Okay. Extremophiles, thermophiles. Therm means heat. They like those hot springs. You've heard of the smokers at the bottom of the ocean when they have really new event from magma? That is the only life process on this planet that's not based on photosynthesis. It's based on synthesis from chemicals spewing up from the inside of the earth. Very hot. The water is way above the boiling point of water. And the organisms are alive. Also, why is the water not boiling? Way above boiling pressure, hydrostatic pressure. Hot water. Sometimes, if you use hot water wash and you use detergents for hot water, they've taken some of the enzymes from some of these thermophiles, not necessarily the ocean vents, but from someplace that's hot. And enzymes work better in hot water. Way back to what we said. These enzymes have the correct hydrogen bonding, they will work in hot water. You move into cold water thinking you're going to save money, what happens to your enzyme? It won't work, because what happens to its shape? You moved out of its optimum temperature. Okay? Halophiles, not only the ocean so much, Great Salt Lake and Dead Sea. You've ever seen evaporation ponds? Do y'all use sea salt? Do you use sea salt? They block off part of the ocean and they let the water evaporate, which is actually pretty good because you get potassium chloride, magnesium chloride, magnesium sulfate. You get not just sodium chloride. For a while, before it completely dries up, these change beautiful colors because the different types of archaea can survive as the salt level gets higher and higher. But eventually they do die. They can't grow in the air. The big one right here, methanogens. That's the hugest group. They have to be anaerobes. They are only anaerobes. That means they can only grow if there is no oxygen. They will change carbonate into methane. You've already sort of heard this before, so this is a little bit of old information. Methane, when given off, will hold heat in something like 40-something times more than CO2 will, if it's not tied up in the sediments in the ocean and in the dirt. Okay? You can also get it from sewage treatment plants, as it says here. Methane is very flammable. Some of them, sewage treatment plants, burn the methane, and therefore they can run their lights or maybe some of their air conditioning. Use it for a source of energy. Yes, it hopefully will stay sequestered. If it comes out into our atmosphere in large quantities, our world will heat up dramatically. However, just to give you a link, because you're going to hear this later, we are a bit protected, besides the fact it's locked in the soils. Remember I said biofilm, different organisms form and they have arrows at the top, arrows at the bottom, back to the arrows throughout. Phenogens are what? Arrows. They are what? Deeper in the sediments. Deeper in the sediments. Said they produce what? Methane. Flammable. Well, what just happened? And this overlaps with the chapter on metabolism. They took carbonate as the electron acceptor, threw electrons on it, which is they had to stop somewhere. They have now loaded this with electrons, which means it has higher energy. It's an energy source, if you're not a fan. Just above them are the methane type of bacteria called the methane oxidizers. Guess what they do? They take the methane in and they use it as a source of energy to run cellular metabolisms. And that's a nice system right there. Does all of it get used up? No. Some methane gets through. But right there is a little bit of an environmental, naturally set up protection. Okay, some of you are giving off methane because some of you have advantages in your intestines. Certain percentage of people do. All cows do. You already heard about the cows giving off the methane because they have symbiotic, meaning the pathogens and other bacteria in their forestomach, the rumen. Cow cannot break down grass. It will starve to death. Only the bacteria break down the grass. And in the process, some of them are methanogens because they're archaea, and they give off methane. Yes, cows poop methane, but they're not going to ruin the world. Any questions on this? Moving to the back, I was trying to draw a volcano because these are some of they think, the most ancient bacteria of all. And when our planet began, there was no oxygen. It was volcanic and it was anaerobic. So these were the organisms that were some of the first. Deeply branching, that simply means they tend to look like they have filaments. And I said about the first ones, think about your carbon energy sources. Photoautotrophs get their carbon energy from light and CO2. Make photosynthesis. We are chemoheterotrophs. We have to have multi-carbon compounds to break down to get pieces of carbon for bricks and energy. Which had to come first? First you have to have the photosynthetic autotrophs. So that's what this is saying, is they had to be some of the first because they can live off of just traces of chemicals, CO2, and light. And then it says Deinococcus is resistant to radiation. That's back to an overlap from our chapter we're finishing up on genetic engineering. They haven't succeeded with this yet. Radiation can cause what? Damage to DNA. For instance, remember we talked about ionizing, mutation. Ionizing radiation breaking the phosphate backbone of DNA. That just tears it apart, doesn't it? Deinococcus is still damaged. He's still very damaged by radiation. But his repair systems, remember our repair systems we talked about? For instance, pyrimidine repair system repairs UV damage. His repair systems are so fast, he fixes the DNA before it has a chance to cause any harm or damage. They haven't found a way to use that yet, but they're thinking about different applications. And I said, this is a lot of, and I'm not going to try to bore you to death, and I'm going to try to get you some pictures and things to help make it more interesting. This organism has this trait, this organism does this. I would make flashcards. I would learn each set at a time, but you're going to have to memorize it. I can't think of any other way to teach it besides just have to know it. Because there's no interactive cycle going on here. There's no processing going on here. The next one I'm going to give out, the next ones, what I'm going to do, I'll take two of these. They say that these are the low G to C base pair ratio organisms. Let me explain what that means. Because it's not all or none. I'm gonna keep doing that. 1, 2, 4. Remember, this is in text format in the focus notes, so if you don't like this, and this is going to change as we get a chance to get to it. I always want to improve things. I'm never happy with what I have. I am never happy with what I have. Yes, I know avoidance. You're looking at the clock. I don't have you happy and engaged. I have to get something that's engaged. Okay? Yes, y'all can look at the clock. That's gonna fall apart. Thanks. I'm not skipping you, I'm trying to.
I thought she was keeping them.
Now, why would you do that, would you? Does everybody have one? Does everybody have one? Okay. No, nobody's keeping. It does happen though. It's like never being at the end of the table when they pass out the food in a large family, you know? Okay. 2 ways to classify bacteria. There is not a way. They can use this, which is the GC-base pair ratio, which we learned way back in chapter 1. Take the bacteria, tear it apart, find out what percentage of guanine, cytosine, adenine, thymine it has, plug it into a formula, and it will have a number below 50 or above 50 or around 50, and they say that's a group of organisms. That only works sometimes for some species. And other species, it makes no sense at all. For the other species, some of them use ribosomal RNA sequencing. For some other species, sometimes they just flat out use the old biochemical test. So yes, I understand that's confusing, but microbiology is just not clean cut. That's just all there is to it. That's what we do. So what I'm going to say is you don't have to know if it's G to C base pair ratio. You don't have to know if it's ribosomal RNA. Just learn the organism, what I tell you about it, and what it does. That's all I'm asking you to know. Because they're going to change it one day anyway. All right. These, I'm going over our 2 spore formers. Who are our 2 spore formers? Bacillus and Clostridium. You are so going to know that for the rest of your life. Okay. Now, Bacillus has peritrachous flagella. That's like perimeter, so it means the flagella go all the way around. It's a facultative anaerobe, can be anaerobe. He does oxygens okay. I tried to break it down into environmental species, species that are pathogens, species that might produce antibiotics. So I at least have some sort of organization to it. Up here in this section are the bacillus species that give us some antibiotics. Some of the bacillus species that can give us antibiotics. There's Bacillus polymyxin. You ever seen that triple antibiotic cream that you can buy? It has 3 antibiotics in it. Bacitracin, polymyxin, and also neosporin, if they can change it a little bit. You never take it internally because those antibiotics will destroy your kidneys. Okay? But for a topical, it's pretty good. It's pretty good. So Bacillus polymyxin simply makes the polymyxin antibiotic. It actually inhibits protein synthesis. We'll teach you that when we get to the chapter on how antibiotics work. Now here is Bacillus lixiformis. Say that 20 times real fast. Bacillus lixiformis. Okay, it's Licheni and then formis. It makes bacitracin. Bacitracin. Again, one of those triple antibiotics. Look at them sometime and maybe bring one in so they can. Bless you. Bless you. Down here is our one that we mentioned over the last chapter, but it's agricultural. Bacillus thuringiensis. Bacillus thuringiensis makes the Bt toxin. And of course that kills insects, caterpillars, and they have done some genetic engineering with that. Moving down here is my section for the pathogen, the worst pathogen. Bacillus anthracis is what? Anthrax. There is a difference in your survival chances versus how you catch it. If you get cut, and it can turn into a black wound, that's called cutaneous cut. You can have maybe 20 whatever percentage chance of mortality. Probably a good chance. If you breathe it in, inhalation anthrax is almost 100% fatal. Way back when the people that were very bad had the bacillus spores and mailed them up to the Senate office up in D.C., some of those letters passed through the mail system in Florida. We had a Florida resident who had to handle that, didn't know what it was, just doing his job. He breathed it in, and he died. It was one of the cases they don't really tell you about. And if you're doing this, you ever seen flour that foofs up? They dry the bacterial cultures and spores to where when you open it foofs out lyophilized, if they do this. And it just goes everywhere. That Senate office is still sealed shut because they can't get it out. If you have spores and you have cement, what does cement have? I don't know what the office is made of, but it would be in the homes. You can't have carpet, can't have ceiling tiles. Every teal, nook, and cranny would have a spore. Okay. Now moving over here to Clostridium. All of it anaerobe. Anaerobe only. Oxygen kills. Spore former. Yes, it can produce toxins. I want you to know the four. Question? No. I want you to know the four species. Clostridium tetani gives you tetanus. Clostridium perfringens, yes, there's overlap with food poisoning with every organism on the face of the planet, but I want you to know Clostridium perfringens for gangrene. Clostridium botulinum is food poison, botulinum. Very bad for that. Okay? Clostridium difficile, you know by the nickname C. diff, massive diarrhea. And probably too much in the way of antibiotics for too long a time. Okay? They're down here. Okay? And if not, they're also the focus ones. A little closely related to the Clostridium by this representative is Veillonella. All I want you to know is he's a gram-positive that can cause problems in your mouth. Okay?
Like the what?
Veillonella, V-E-I-L-L-O-N-I-A. V-E-I, like in veil. V-E-I-L, bless you. Bless you. L-A-N-I-A. It's an anaerobe too. It can help cause cavities, just know that. Dental plaque. Dental plaque can turn to harden, and you can eventually get cavities and your tooth is in trouble. Mycoplasmas break everything I've ever told you about bacteria. Everything I've taught you about bacteria, they break the rule. They're the smallest single life form that we know of at this time, as viruses don't count. You ever taken an egg and you drop it down? You have the yolk sitting up and you have the white that sort of spreads out. They on a plate will look just like that, like an egg. The colony looks like an egg dropped down on a plate. Not yellow and white, but up in a halo. They need almost every nutrient you can think of. They're osmotically fragile. And in fact, they have no cell wall. They're bacteria that have no cell wall, which means they have no peptidoglycan. What have I been teaching and teaching? All bacteria have peptidoglycan. This is the exception. Okay? This is the exception. Now, that makes them osmotically fragile. Remember all the talk about osmotic pressure? Water flows naturally in. If the cell membrane doesn't have a cell wall to come up and stop against, it tends to lyse. So they have to be in places in your body where they're in an isotonic environment. That means about the same number of solute particles outside as inside. They like your cell membranes. They like your cell membranes because you have some dissolved different types of things in there, and it helps equalize out the osmotic pressure so they don't blow up or lyse. And I'm out of time. Any questions? Yes?
Mycoplasmas are they also anaerobic?
On that.